Hello everyone, welcome back. We are looking at revisiting albinism, and we're now looking at how genotype and phenotype are related. Recall, genotype is the combination of genes that you have on your chromosomes, and these come from your parents, they're on your chromosomes, and ultimately whatever combination of genes you have, that will determine the physical trait or phenotype that a person happens to have. At this point, you should be completed on the second page with this chart and these three questions. So I'm going to be picking up today by looking at the activity that we're going to be looking at, which is kind of going through genotypes and how that determines phenotypes. Before we get there, I want to touch base with you relating this codon wheel, as it's called. So let me explain really quickly. Some things we need to know. DNA ultimately is used to produce proteins. Proteins are the things in our body that really determine everything. Our hair color, eye color, all of it is controlled by proteins, which ties back to DNA. So in order for DNA to actually be functional, it has to be turned into a protein. Proteins, they're made of tiny molecules known as amino acids. So when we look at this chart up here, all of these funky words, those are all amino acids. So arginine here, that happens to be an amino acid. So what we now have to consider is what combination of DNA will ultimately code for this amino acid. And also know we have to string together many amino acids to create one protein. So a protein would be one, and in amino acids we gotta have to link together many. So one of these is going to equal many of these. And as we start to go through this, you'll kind of see what I mean by that exactly. Now looking up here, we start to see some of the letters that we're familiar with seeing in DNA. We have G, A, and C, but we notice that there are no T's. Instead, we have U's. In order to create an amino acid, we have had to change DNA a little bit. DNA started as DNA, but in order to create an amino acid sequence, DNA has to be converted into RNA. So DNA, it is changed into RNA. And RNA can then be changed, essentially, into amino acids, and that leads to the creation of the protein. So we've been talking about many of these things already, we just haven't really talked about these two. We kind of went straight from DNA to proteins, but we overlooked some of the middle steps. So how do we get RNA from DNA? Well, it's quite simple. In our handout, I'm going to start by demonstrating using some of the assignments that you guys will actually be doing. In your handout, you'll see 4 of these. Here we have one gene, TYR, which was mentioned in our last video, and the OCA1. On the next page, you would have OCA2, and so on and so forth. Going back to TYR, this gene, here's our DNA strand. We need to change this DNA strand into an mRNA. All we do is the exact same thing that we had been doing for DNA to create the complementary strand. A goes with T, C goes with G, so on and so forth. However, in mRNA, which is a form of RNA, the nucleotide T, it does not exist down here. So anywhere you would want to put a T instead, we're going to be putting the letter U. So what bonds with T? A. What bonds with A? Well, it would be T, except T does not exist in mRNA, so it would be U. What bonds with C? G. So we continue in similar format to create our mRNA strand. See, we got G, so we have C, we have A, there's no T, so there'd be U, G, C, C, U, G, G, C, U. I'm going to pause this video and kind of fast forward a little bit while I fill in the rest of this. So I've completed my mRNA sequence. I used my DNA strand as a template. Now I can use the mRNA codon wheel to figure out which amino acids this particular strand of mRNA will happen to create. So to do that, I have to go back to my codon wheel. Here we are. Now how do we read this thing? This in here shows us the first letter of a potential codon, which, by the way, a codon is a 3-letter sequence of mRNA. So here we have a codon. This also is a codon. This also is a codon. Once again, a codon is a 3-letter sequence of mRNA. The innermost letters that we see in this wheel indicate the first position of a codon. That will make sense in a minute. The next tier out will be the second letter in a codon, so on and so forth. And then the outer ring would be the third letter in a codon, so on and so forth. So let's check this out. My first codon is AUG. What amino acid will that create? So let's find our first letter A, our second letter U, our third letter G. So AUG looks like methionine is my amino acid. I'm just going to write the first 3 letters of that, MET. Most amino acids, you just use the first 3 letters as you kind of fill them in. So MET. My second codon is CUC. So first letter C, second letter U, third letter C. I have leucine. So I'm going to be writing LEU. Now, with leucine, something that you should see, which was coded by CUC, that 3-letter combination creates leucine. But also, what if this was CUU? CUU. A different set of 3 can create the same amino acid. What about if this was CUA? CUA. Still the same amino acid, and hopefully you're picking up CUG would still code for the same amino acid. So I could have, in this scenario, 1, 2, 3, 4 different possible amino acid sequences, and I would get the exact same amino acid. So sometimes different codons will create the exact same amino acid. Let's go do a couple more here. My next one is CUG. So CUG, looks like I got leucine again. And next one, GCU. GCU, looks like I got ALA this time. And I'm going to continue on down. I'm going to pause the video while I fill this in. So my completed strand of amino acids is now complete. I can see that there's a lot of amino acids in the particular gene TYR, creating oculocutaneous albinism. This strand right here of amino acids is now no longer one strand of many amino acids. This whole thing is one single protein. And once again, this protein will control albinism. Now, do understand, going back, that this amino acid sequence I just created, it is my original strand, meaning that this is how it should be. There are no mutations. But there's different types of mutations that can occur that will change this protein. So what you will need to do with your group members is complete 3 different mutations that could happen to this particular protein. Furthermore, you all will be not working on the exact same strand of DNA. One person in your group will be continuing on with this one, working on the TYR gene. A second person in your group will be doing the exact same thing that we've done here, but for the second type of oculocutaneous albinism caused by a different gene. Once again, same thing. You'll have to create the mRNA strand for the original normal unmutated strand, and then come up with the list of amino acids creating that protein. But then you will have 3 mutations that you'll need to be looking at as well. You will notice in the mutations that the sequence of amino acids in the original strand, they are not the same as in the mutated strands. So you'll be asked to look through that. So take a moment, get your particular page filled in, and then you'll be asked to trade with a neighbor. And then you guys will really only be doing 1/4 of this. So we have a second gene, a third person group will be doing the third type of albinism, and a fourth person group will be doing the fourth type.